Welcome to LankanTutor, guys. This is Biology 2.3, where we'll be looking at organic molecules, specifically carbohydrates. Before we move on to carbohydrates, let's look at what an organic molecule is. There are four major types of organic molecules found in the human body, or any living organism for a matter of fact, and they happen to be carbohydrates, lipids, proteins, and nucleic acids. They're called major biological molecules because they are found in all living matter. Some of them are macromolecules and some of them aren't. What exactly is a macromolecule? A macromolecule is a very large molecule, specifically with a molecular weight of 10 to the power 4 to 10 to the power 10. Now, you're going to have to remember those numbers because they actually ask you the molecular weight of a macromolecule. And a macromolecule are built up of repeating units. These units are called monomers. One thing you need to remember about the biological molecules, carbohydrates, lipids, proteins, and nucleic acids, the only one of this which isn't a macromolecule is lipids. So remember that lipids are not macromolecules. That's going to come back again and again and again. So make sure you remember that lipids are not macromolecules. All macromolecules are polymers. Again, polymers are made out of repeating units called monomers, like we said before. And there are some features of macromolecules you guys should know. When these repeating units or monomers bond together, they remove water. This is called a condensation reaction, where two hydrogen atoms and an oxygen atom remove and the bond is formed between the two monomers. You will see that in the diagrams that I'm going to show you when we're going in detail about carbohydrates and everything. So don't worry about that just yet. You have a few more minutes before we get into those structures. And again, bonds between the subunits, when they're broken, water is removed. This is called a hydrolysis reaction. And formation of bonds require ATP, and it is an anabolic reaction. The difference between the two types of metabolic activities, anabolism and catabolism, how anabolism requires ATP and catabolism generally releases ATP. You can look at 2.1 if you want to, and if not, let's continue this. So you have a general idea of what an organic molecule is and what a macromolecule is. Since we know the four types of organic molecules, we're going to go into detail of each and every one of them, starting with carbohydrates. Everyone should know what a carbohydrate is. In the food pyramid, carbohydrate is the base or the staple type of nutrient that we consume. But that's not what we're searching for. We're searching for the molecular structure and what carbohydrates actually are. So you need to remember that carbohydrates are the most abundant organic matter on the earth. That's right, they're actually more common than nucleic acids and proteins and lipids. The major elemental composition is carbon, hydrogen, and oxygen. Remember, all organic compounds contain carbon, hydrogen, and oxygen. To be considered an organic compound, it has to have those three elements. The general formula of carbohydrate is CxH2OY. H2O ratio is always 2:1, just like water. Remember that X does not always have to equal Y. Let me give you an example. C6H12O6, that is the common formula for glucose, and C12H22O11. You see that Y can be X or not. It doesn't necessarily have to be that, but just remember the formula CxH2OY. We know what a carbohydrate is. You should be able to give a brief description by now. It is the most abundant organic matter. It comprises of CHO in the general formula of CxH2OY. And the ratio of hydrogen to oxygen is 2:1. Carbohydrates are divided into three main categories: monosaccharide, disaccharide, and polysaccharide. Now, when I say these names, you should be able to figure it out. A monosaccharide has only one saccharide. Disaccharide means two, and poly means many. So what exactly is a monosaccharide, and do you know any of these? Monosaccharides are simple sugars. They're the basic types of carbohydrates. Many monosaccharides get together to form polysaccharides, or two of them get together to form disaccharides. Even the monosaccharides get classified again, depending on how many carbon atoms they have. Let's take an example of a triose. Remember, you're going to have to memorize all these examples and remember all the types of monosaccharides. First off is a triose. Trioses have 3 carbon atoms. For example, take glyceraldehyde. Glyceraldehyde is an intermediate in respiration, and it is also a raw material to synthesize many cellular compounds. And phosphoglyceraldehyde is a derivative of the first product of photosynthesis, and it also serves as a major raw material for synthesis of carbohydrates. But don't worry about that, we'll get into photosynthesis later. Remember that trioses have 3 carbon atoms, and examples for those would be glyceraldehyde and phosphoglyceraldehyde. And next we're going into 4 carbon atoms, or tetrose. Now, the thing you need to remember about tetroses are that they don't occur in the environment that often. It's very rare, so you don't really have to learn a lot about it, but you need to know that the example that we need to remember is called erythrose. So erythrose is a 4 carbon compound, or a tetrose. And then we move on to 5 carbon compounds. These are really common ones that everyone knows, but just remember that they're pentoses, and those are ribulose, ribose, and deoxyribose. Now, ribose is a constituent of RNA. RNA is a genetic material found in organisms and viruses and such. Ribulose is a component of a carbon dioxide acceptor called RUBP, or ribulose bisphosphate. Deoxyribose is the infamous constituent of DNA. So you might want to remember that one, and it's kind of hard to miss out. The next is a hexose, and hexoses are the most common types of carbohydrates, at least the ones that we know about, because they happen to be the sugars like glucose, galactose, and fructose. Even though glucose, fructose, galactose, they all have the same amount of carbon atoms, how would you recognize what the molecular formula is? For example, I'm going to draw these right now, and you should know that the general formula for glucose is C6H12O6. That means it has 6 carbon atoms, hence it's a hexose. But galactose and fructose also have 6 carbon atoms. They're also C6H12O6. So what is the way you can identify the difference between a glucose, a galactose molecule, and a fructose molecule? This feature is called structural isomerism. You'll learn a lot more about it in chemistry, but what basically happens is the carbon atoms, they all have the same amount of carbon atoms, but the location of the carbon atoms and the ketone and aldehyde groups are different. So those small differences are what separates a glucose from a galactose and a galactose from a fructose. Now, we know pretty much everything we need to know about monosaccharides, but then things get complicated. Monosaccharides combine with other monosaccharides to form disaccharides. Remember when I said this anabolic process requires an ATP and it occurs through a condensation reaction, meaning the water molecule between these two get removed. Specifically, there are 2 OH groups on either side. One OH group gets completely removed, and another hydrogen atom from the other OH group relieves and forms the bond. This bond is called a 1,4-glycosidic bond. The reason that it's called a 1,4-glycosidic bond is because of the location of the carbon atoms. It's between the first and the fourth. It's between the first carbon atom of one molecule and the 4th carbon atom of the other molecule. There are also glycosidic bonds called 1,6-glycosidic bonds, but that happens in a completely different scenario in polysaccharides when it'll be between the first carbon atom and the 6th carbon atom, and that happens when there are branched chains. If it's a linear polymer, there is going to be no 1,6-glycosidic bonds. Now, remember that these reactions are also reversible. If you have a dilute acid or some enzymes, you should be able to break the disaccharides into monosaccharides again, and hydrolysis takes place. I said that the disaccharides are created from the union of two monosaccharides, specifically the 6 carbon monosaccharides like glucose, galactose, and fructose. An easy way to remember how these monosaccharides join is to remember that glucose is needed in every one of these. Disaccharides cannot be formed without glucose. So let's write down glucose on all 3 scenarios here. Remember, there are 3 types of disaccharides. Glucose gets together with another molecule of glucose to form maltose in the presence of an enzyme called maltase. It's really easy to remember. All enzymes have A's, and maltase, maltose, you can figure that out. The next one is glucose is joined with a galactose molecule to form lactose. Lactose is commonly referred to as milk sugar. Maltose is commonly referred to as malt sugar. Now, glucose and fructose is the type of sugars that you can find in fruits, join together to form a very common substance called sucrose. Sucrose is the white table sugar that people use in cooking and everything, and it's commonly called cane sugar. Now, if you want to find out if these sugars are present in a sample, let's say, there are 2 tests that you could take. One is using the failing solution and the other is using the Benedict solution. Now, these solutions are alkali copper sulfate solutions, and when they're boiled with reducing sugars, the Cu2+, the copper 2+ ions, are reduced into copper plus ions, and a brick red precipitate is formed. It's called cupric oxide. All monosaccharides are reducing sugars, and all the disaccharides except sucrose are reducing sugars. So every reducing sugar without sucrose, when you use this test, you will get a brick red precipitate. So this is how the experiment is done. 2 milliliters of reducing sugar, be it glucose, galactose, fructose, maltose, or lactose. So 2 milliliters of reducing sugars, that means any mono and disaccharides besides sucrose, is heated up with 2 milliliters of, let's say, failing solution, and it's brought to a boiling temperature. The first thing to remember is that it starts off blue and then it turns green, and eventually, if it shows a presence of reducing sugars, a brick red finally precipitates. And if it doesn't show any precipitate, that means there are no reducing sugars. But how do we test for sucrose? Because this is not going to work. Sucrose is not a reducing sugar. And if we do that, it'll give you the same example, the same color as it would if you used a non-reducing sugar or something which is completely not a carbohydrate. Again, if you guys remember a few minutes back, I told that disaccharides could be broken into monosaccharides by the use of enzymes or dilute acids. So what you need to do, so if you've done the Benedict's or failings test on a sample and it gives green with no brick red precipitate and you believe that it has sucrose, what you could do is you could add a dilute hydrochloric acid or an enzyme, but let's just leave it at something more practical, which is dilute HCl. You can add it for a little while and the sucrose breaks into its monomers. So those monomers happen to be glucose and fructose. Even though sucrose is not a reducing sugar, glucose and fructose are reducing sugars. So after a little while and you boil it again, it turns out as a brick red precipitate. So the only way to find if sucrose is present is by breaking the sucrose disaccharide into glucose and fructose and testing that for reducing sugars, which should give you the answer. So we've done a lot about monosaccharides and disaccharides. Let's move on to the final part, which is polysaccharides. Polysaccharides are formed when there are a bunch of monosaccharides, more than 2, that form together to create extremely large molecules. Now these molecules can be linear or they can be branched. If you remember that disaccharides are linear and monosaccharides are, well, just one molecule. All of the polysaccharides are polymers and they are macromolecules. They are made out of repeating units and they are rather large. So let's get into a few examples of polysaccharides. Remember, you need to remember all these examples because this is a big unit, and they've actually asked this essay a lot to explain the structure and function of carbohydrates, or at least just the structure if it's a short note. You have half an hour to write all this stuff down. It shouldn't be so hard. I'll tell the points that are needed at the very end, so you can basically skip through all the unnecessary details if you've already done this lesson. Starch is a storage polysaccharide which can be found in plants. Starch has two components, amylose and amylopectin. Amylose is made out of 1,4 glycosidic bonds. That means it is a linear structure and it does not have any branches. So amylose is just like a long spiral of glucose monomers. Now you need to remember that the monomers of starch are glucose. Amylopectin, however, is made out of 1,6 glycosidic bonds as well. So it produces large chains and it makes it much more complicated. So starch is a storage product in plants, and we're going to move on to a storage product in animals, and it's going to be glycogen. It is a storage polysaccharide in fungus and animals like humans. So we're going to move on to glycogen. It is a storage polysaccharide in animals, unlike amylopectin. It's also found in fungi. Now glycogen is created in the liver in small granules. Don't worry about the liver, we have a whole section based for that. It's much more branched than amylopectin. It has far more like 1,6 glycosidic bonds, and it is a generally larger structure. Because of this makes glycogen more soluble than starch, and glycogen can be hydrolyzed by enzymes which will be broken into glucose. So again, starch has glucose monomers and so does glycogen. Then we go into structural polysaccharides. For example, cellulose. Cellulose is the most common structural polysaccharide or most common organic molecule on the planet because it is the structural component of plants, and there are a lot of plants out there. All the plant cells are created with materials like cellulose. A single cellulose chain can contain around 10,000 sugar units, so they're rather large. The cross links between the adjacent chains, it's by hydrogen bonds. It makes it rough. These fibrils are never branched. You need to remember that they do not have 1,6 bonds. They are 1,4 linearly arranged, humongous structural molecules. The difference is cellulose is a completely permeable structure which allows water to go in and out of cells. That's how water is transported within the plants, because cellulose is permeable. Otherwise plants cannot function. Cellulose is another polysaccharide which is made out of glucose monomers. The reason that people like us can't really eat grass or eat the stem of a tree is because we can't digest cellulose. The difference between us and herbivores is that herbivores contain these parasitic organisms or these weird forms of bacteria inside them that secrete cellulase. These organisms living in the gut secrete cellulase, which helps animals digest the cellulose and get nutrients. So the reason that people can't live off eating greens, when I say greens, I don't mean cooked vegetables, I mean raw grass. That's because we don't have any way to digest those cellulose. We are at the fifth polysaccharide, which is inulin, which is again a storage polysaccharide. But finally, for a little change, this is a polymer of fructose. Then we're at chitin. Chitin is a structural polysaccharide. It's commonly found in the exoskeleton of insects and fungi cell walls. It's kind of like cellulose, except the cross-links are based on nitrogen, and it is a nitrogen-containing polysaccharide. Now remember, I said that polysaccharides have to have carbon, hydrogen, and oxygen. They can have other elements as well. They just can't not have CHO. Next, we move on to hemicellulose. It is a pentose polymer, which means that it is formed from 5 carbon monomers. It is a structural polysaccharide present in plant cells. So the 8th polysaccharide is pectin, and it is a structural polysaccharide present in plants. And the monomers here are galactose. And then we move on to murine, which is present in bacterial cell walls, and it is cross-linked with amino acids. So let's divide the polysaccharides we got. There are 2 types, one structural and one functional. The structural ones are cellulose, chitin, hemicellulose, pectin, and murine. The functional ones, in which the function generally is storage, is starch, glycogen, and inulin. So let's just wrap this up for today. Before we leave, let's do a quick recap on what we've done. First of all, carbohydrates are divided into 3: monosaccharides, disaccharides, and polysaccharides. Monosaccharides and disaccharides are sugars. Polysaccharides are not sugars. The mono and disaccharides have small molecular weights. They are not macromolecules. Remember that monosaccharides and disaccharides are not macromolecules. They are sweet and they are readily dissolvable in water and they crystallize. Polysaccharides are macromolecules with large molecular weight. They are not sweet and they are slightly soluble in water and they do not crystallize. All monosaccharides are reducing sugars and all disaccharides except sucrose are reducing sugars. And finally, we're going to the functions of carbohydrates. Carbohydrates with storage functions are starch, glycogen, and inulin. The structural happen to be cellulose, chitin, and pectin. And in monosaccharides, glyceraldehyde is an intermediate in respiration. Phosphoglyceraldehyde is a derivative of photosynthesis. Ribose is a constituent of RNA, ATP, and coenzymes. Ribulose is a component of ribulose bisphosphate, the carbon dioxide acceptor. Deoxyribose is a constituent of DNA. Glucose is a respiratory substrate, and sucrose is a storage carbohydrate in cane sugar, and it involves in phloem translocation. There you have it, guys. This lesson was rather long, and don't worry, the others are going to be just as long because now we're actually getting into the hard parts of biology. So thank you guys for watching. If this video did help you out, don't forget to like, comment, share, and subscribe. If you're not watching this on LankanTutor.com, make sure to do so because it has some tutorials which can help you. So until next time, have a great day. And if this video helped you, don't forget to share it with at least one of your friends. Until next time, see you guys later.